I'm going to talk about the circulating signals of ME/CFS. This is a title that's somewhat shorter than my title in the program, as I'm not going to really talk a lot about exercise challenge. So I'd like to start by introducing my lab members here in this photo. I'm going to be talking only about a small subset of the work that's going on in my lab. The individuals pictured here, this photo was taken last summer. One of those individuals, Jessica Maya, has now left and is working as a postdoc at the CDC. And two students have now joined my lab who aren't pictured. We have to get a new photo when the weather gets better. So our center is located in this building, a part of the center, my lab and Andrew Grimson's lab, and we have several other collaborating labs. If you want to learn more about our center and its research, I encourage you to go to this website I'm showing here, neuroimmune.cornell.edu, and you can also find a number of talks that either I or other members of the center, or my students or postdocs have given at previous meetings. So I decided I would talk today about the question of a persistent antigen. This has been getting some publicity in the field of ME/CFS. And after hearing the last talk, you might think this is something that may not be reproducible, but what I'm going to be telling you today, that a number of different groups are now looking at this question of persistent antigen, and there's actually some reproducibility. Although I agree with the previous speaker that there is a great deal of heterogeneity in the patient population. So a persistent antigen is one that remains in the body for a long time, and it's continuously stimulating the immune system. These persistent antigens are known to occur in chronic viral or bacterial infections, and some classic examples are people who are infected with hepatitis B or people who have tuberculosis. So these antigens are often proteins or parts of proteins that are stimulating the immune system that arise from these pathogens. There are also autoantigens, which are human proteins that are being erroneously recognized by the immune system, and tumor antigens, which certain proteins are highly expressed by tumor cells, and those are also agitating the immune system. So this persistent antigen stimulation can result in something called exhaustion of the immune system. And this phenomenon of exhaustion is a specific phenomenon. You have to have certain changes in gene expression and in the function of immune cells to conclude that exhaustion is actually happening. So I think most people in the audience are probably familiar with this paper that recently appeared. This is the long-awaited work of the NIH intramural program. And one of the conclusions that they made was, in fact, that there was a persistent antigen. This is a quote from them: "The cause of immune dysregulation is not clear, but may suggest the possibility of persistent antigen stimulation." Notice that they say may suggest. That's because they don't have a great deal of evidence for the presence of exhaustion, but they do have some evidence, and I'd like to describe that. So what was this evidence in that paper about chronic antigen stimulation? So first of all, they show in the paper that there was an increase in naive B cells and a decrease in switched memory B cells in circulation. So in their paper, they used red for the ME/CFS subjects and blue for the healthy individuals, and we do the opposite. I just want to point that out so you don't get confused when I start showing you ours, but we actually use blue for ME/CFS when we're showing our graphs, because blue is the ribbon color for ME. But in any case, here you'll see that there are more naive B cells in the ME/CFS population, and there are fewer B cells that are switched. And that means there are fewer functional B cells, which is not a good thing, since B cells are responsible for antibody production. They also reported an increase in circulating naive T cells. They saw this in females, but not in males, and that is actually another source of heterogeneity that we also see ourselves, that there's a lot of sex differences in ME. And finally, they also showed that there was an increase in CD8 T cells expressing a famous inhibitory receptor called PD-1 in cerebral spinal fluid. So this is also another hallmark of the exhausted state. But the conclusion that they made had to be very cautious because they don't have a great deal of evidence for the T cell exhaustion. And so this is what they wrote, that they did find immune dysfunction affecting the immune system, and they found that this immune dysfunction is sex dependent. And the reason this is written this way, we hypothesize that these changes are driven by antigen persistence of the infectious pathogen, is that they don't have direct evidence for the antigen persistence, especially they don't have evidence for it being the infectious pathogen that is remaining. But nevertheless, it's a plausible hypothesis for them to make and to point out that their data is consistent with this hypothesis. I believe that probably they felt reassured in making this hypothesis from the fact that in long COVID, there's actually much better evidence that the infectious pathogen can persist and cause disruption of the immune system. But what could be the source of a persistent antigen in pre-2020 ME/CFS? It's certainly not SARS-CoV-2, but it could be an undetected chronic infection by an inciting virus. And an example of this is an enterovirus. In previous talks, which in fact you can find on on our center website, I described the evidence for enteroviruses in ME as the inciting virus. And also I described the fact that enteroviruses are known to be able to persist in tissue after the acute infection is over. It's also possible that a persistent antigen could come from a known chronic infection by herpes viruses. And the most prominent one of these is EBV. And this could happen especially if the immune system has lost control of these viruses and they're reactivated. Another possibility is that there could be some abnormal expression of endogenous retroviruses, or there could be a normal human protein that following an infection has become recognized by the immune system. And I believe in the next talk, we're going to be hearing about that possibility from Carmen Scheibenbogen. So what are the previously known immune system abnormalities that suggest stimulation by a persistent antigen? I think this is one of these papers was cited in that NIH intramural paper, and it probably also gave them some reassurance that their hypothesis might be correct. So in 2020, we published a paper about T cell metabolism and another paper in 2023 about fatty acid oxidation in immune cells. So let me just mention that T cells normally alter their metabolism to respond to foreign antigens. And two of the primary types of T cells are CD4 T cells and CD8 T cells. So CD4 T cells have an important job to signal other immune cells to respond to an infection. The CD8 T cells, which I'll be talking about a lot, are ones that secrete cytokines, but are also very important to cause death of infected cells or, for example, of tumor cells. Following stimulation of the T cell due to detection of a foreign antigen, T cell metabolism normally changes. So in a quiescent T cell that doesn't know that there's a pathogen around, typically in a healthy individual, that T cell is using fatty acid oxidation. But after the cell has received a signal that there is a pathogen around, the cell will upregulate glycolysis and oxidative phosphorylation. It'll put its mitochondria to work to battle the invading pathogen. Now, the problem is that when the immune system is being chronically stimulated by a persistent antigen, these metabolic responses do not occur properly. So in our study in 2020, we showed that glycolysis was lower in ME than in healthy controls. we also showed that the mitochondrial membrane potential was lower in ME versus the controls, meaning that the mitochondria weren't as functional as they should be to carry out oxidative phosphorylation. And in our more recent study, we showed that fatty acid oxidation was higher in ME than in the control. And, bearing on the point of heterogeneity, you can see that it looks like there's some, at the bottom there, some ME patients that look more like controls, while there are others that look like they have much higher fatty acid oxidation. So this wasn't the only report that is consistent with stimulation with the persistent antigen. So another report appeared in 2023. This report came from Lisa Seeling's lab. And what they investigated is this phenomenon, and that is when a foreign antigen is detected, CD8 T cells receive signals to produce certain cytokines to aid in defense. So the cytokines that these CD8 T cells produce are interferon gamma and TNF alpha. But this response might not occur if the CD8 T cells are dysfunctional due to a persistent antigen. And in fact, that's what they found. So here's a figure from their paper. They found that fewer CD8 T cells from ME and long COVID subjects responded to stimulation with the production of these cytokines, interferon gamma and TNF alpha. So here, this shows what happens in healthy individuals. There's lots of production of interferon gamma, but in either long COVID or in ME/CFS, there is much lower production of interferon gamma. The same is true for TNF alpha. Here we have the healthy controls, and in either long COVID or ME, there's less production of TNF alpha. And that's a hallmark of disruption because of T cell exhaustion, although it's not sufficient to prove T cell exhaustion. Now, I've talked to you about CD8 T cells, which is a subset of cells, and one thing that I've emphasized in previous talks is that it's really important to analyze specific types of immune cells. One source of irreproducibility, I believe, is when you analyze all white blood cells together, you can miss things that are happening to particular individual types of immune cells. And so the paper I want to describe now, which was spearheaded by Andrew Grimson's group, was one that had the most extreme example of looking at individual cell types. And they did the only report that I know of single cell transcriptomics in ME/CFS, where individual cells were examined. So you could look at all the different types of immune cells. So they did single cell profiling of immune cells in ME/CFS versus sedentary controls. And this is another point that I like to emphasize. One of the problems perhaps with reproducibility is the nature of your controls. Who are you comparing your ME patients to? For this study, we really wanted to have sedentary controls so that we're matching the exercise level of the ME patients who can't exercise significantly with controls who don't bother to exercise significantly. So in this, Andrew Grimson's lab used the 10x genomics technology, in which you can isolate individual cells, obtain them one at a time, isolate the RNA from an individual cell, and sequence the RNA that is present in that individual cell. And so 30 ME cases and 30 controls were used in this study, and about 4,500 cells from each subject were sequenced to obtain their RNA. Now, the single cell sequencing data can then be analyzed to find out the type of cell whose RNAs were sequenced. So you can identify which cells, for example, were monocytes or B cells or T cells, and then cluster them together. Now, this is a very simplified diagram. I'll show you what the actual diagram, which is generated by an algorithm, looks like. Each one of these dots in this diagram on the right is an individual cell's transcriptome that then is clustered to show, and you can then identify by the particular RNAs that are being expressed which type of cell is present. So up here we have the monocytes. On the left, we have some T cells, CD4 T cells, CD8 T cells, or say B cells, NK cells. So you can cluster all the different types of cells and then analyze them as particular type of cells in this experiment. So then the RNAs present in one type of cell from an ME subject can be compared to the same type of cell in the healthy sedentary control. So what was interesting is that the type of cell with the most gene expression differences were classical monocytes. So here with the blue circle, you're seeing that this group of cells, which belongs to the classical monocyte group, according to the analysis in that previous slide. And the other thing you'll see, this is the only comment about exercise. This study actually used blood from people who, before they did an exercise, and then 24 hours after they did a maximal CPET. You can see there's not actually a whole lot of difference in the differential gene expression before and after exercise, because that, in fact, there was already difference even before the patients exercised. So to tell you the bottom line from that analysis of the classical monocytes, we found that the ME subjects expressed genes indicating they're being signaled to become macrophages. So monocytes are in the circulating blood vessel, but they can be signaled to go out of the blood vessel and into the tissue, become a macrophage, and then go into that tissue to find a cell that's expressing a persistent antigen, like a virus, and then destroy that cell to prevent the virus from getting out and doing bad things. So after finding this in the single cell data, in order to confirm this, what they did was to obtain a larger number of RNA sequences by purifying classical monocytes, in this case from 4 cases and 4 controls, and then sequencing a much larger number of cells to get more RNA, and then looking to see what are the most dysregulated genes found in that larger population of cells. And it turned out that the most dysregulated one was one called GM-CSF, which is a cytokine that stimulates cells to produce granulocytes and monocytes. And the other 2 top dysregulated genes that are higher in the cases and controls is these chemokines, which are telling the immune cells to go through that blood vessel and get into tissue to, in fact, battle against the persistent antigen. So what's interesting is this is reproducible. This is something that's been reproduced. If you look back at other literature, Montoya in 2017 showed that the more severely ill you were, the more GM-CSF you had. And in fact, in 2023, in a collaboration with Columbia, we found that the worse score you have in physical function, the more GM-CSF you would have in your plasma. So in addition to what I've just shown you, which is all published, and you can find this online, we're continuing to analyze that data set from the single-cell RNA sequencing. And the single-cell RNA sequencing, as I showed you earlier, although the classical monocytes were the most dysregulated ones, there was also dysregulation in the T cells. And David Yu in the Grimson lab has now been analyzing the NK and the T cell data from the single-cell RNA-seq to get further information from it. And this is one of his unpublished figures here, working on a manuscript now, that shows that there are a number of differentially expressed genes in each type of these cells, the NK, CD4, a certain type of T cell. But again, the CD8 T cells are particularly dysregulated. They are showing a lot of differences between patients and controls. So we want to look a little closer again at these CD8 T cells. And in order to look closely at this, the CD8 T cells were isolated so that 2 types could be analyzed more thoroughly. So-called naive cells versus cells that have been stimulated by something like a persistent antigen, to differentiate. So a naive cell is one that hasn't seen an antigen, but after seeing an antigen, T cells then develop into what's called effector cells and then memory cells that are actually the ones that do the action on invading cells with persistent antigens. So these CD8 T cells were isolated again in bulk to do deep RNA sequencing, starting with PBMCs and of 7 cases and 7 controls. These were put through flow cytometry to isolate naive T cells and antigen-experienced T cells. And what was found was that the experienced T cells were showing more differences in their gene expression between patients and controls than were the naive cells. So what are some of the differences in these experienced cells that have seen an antigen? Of course, what we're interested in and found is that there were some genes associated with an exhausted state that were upregulated in ME/CFS antigen experienced cells. So 2 membrane proteins called SLAMF6 and SLAMF7, as well as a transcription factor called EOMs, were upregulated in the patients versus the controls. And also, David found that there was dysregulation of genes encoding metabolic enzymes, and this is consistent with our prior finding of lower glycolysis. So there was a lower enzyme called hexokinase, which is needed for glycolysis in the ME patients, and there was upregulation of a protein called GLUT1 that is also known to be upregulated in exhausted T cells. So all of this gene expression data is consistent with T cell exhaustion. At the end of my talk, I want to talk a little bit about work done by Jessica Maya when she was in the lab. She analyzed 15 patients and 14 controls. She isolated CD8 T cells, not by flow cytometry this time, but by magnetic beads, and then put those CD8 T cells into flow cytometry to examine SLAMF6, that exhaustion marker PD1 I told you about before, as well as this TOX marker, which is a transcription factor. And just briefly, I want to explain what a transcription factor, how a transcription factor drives exhaustion. So transcription factors are responsible for regulating how DNA, which DNA is going to produce RNA. And when the transcription factor TOX is around, it downregulates the amount of RNA that's produced to, say, make cytokines and other immune responses. So you have a decreased immune response in the presence of this TOX transcription factor. It also upregulates things like PD1, which is, again, that classic exhaustion immune suppression marker. So what she found is that exhaustion markers are elevated in cases versus controls in these total T cells. So on the left, we have PD1, which is higher in the ME patients than in the controls. We have SLAMF6, which is also higher in the ME patients versus controls. And of the T cells that are carrying PD1, again, more of those T cells that have PD1 also have this SLAMF6 marker. And we also see that this exhaustion-associated transcription factor TOX is higher in particular populations of maturing T cells. So the TOX is higher here, and of the T cells that are expressing PD1, the TOX is also higher in the ME patients. So this is all consistent with the possibility of exhaustion in ME. So I want to end by summarizing what I've told you about using a slide from a review that was published in 2018. So this slide is slightly modified, but I want to show you that I've demonstrated to you that we and other labs have evidence now that is consistent with the state of exhaustion in T cells. So, for example, we have shown that glycolysis is lower in ME, that fatty acid oxidation is upregulated, that the mitochondria are depolarized, that there's higher GLUT1 in ME than in controls. There's higher PD1. There's higher SLAMF6. I didn't describe increased reactive oxygen species, but other groups have seen reactive oxygen species. I also described that there's less interferon gamma and TNF alpha in the patients versus the controls. So all this is consistent with the antigen persistence that leads to T cell exhaustion. So if there is a persistent virus, the fact that the cells are exhausted could be one reason why it's not being cleared from the body. So what can be done by this? It turns out that a number of cancer drugs have been developed to reverse the exhaustion of immune cells. And the reason that these have been developed is to help the T cells in someone's body to fight against the tumor cells. So this is a potential treatment in ME that should be tested, I believe, in clinical trials. This has to be used with caution, however, because the reason that exhaustion in T cells developed, probably evolutionarily, is to actually tamp down the immune system so that you don't have damage from the immune system being overactive, as has happened when people had acute cases of SARS-CoV-2, for example. So with that, I'll end and just thank the people in my lab who were primarily responsible for the work I presented, although we have many other projects ongoing by those other individuals I showed you at the beginning. Andrew Grimson's lab, the individuals who were particularly responsible for the work I presented. Jen Grenier's lab did a lot of the bioinformatics. We couldn't have done this without the subjects who volunteered to have their blood drawn and even sometimes do a CPET. We had a large number of people involved in collecting samples. And then the funding shown in red is the primary funding we used for this particular study. So thank you very much.
Thank you. Thank you, Dr. Hanson. Will you mind just a couple of more minutes? I'm going to give one question to the audience, okay?
Okay.
A question? We have one question. Thank you.
Kind of experience where the block of SLAM or PDI may revert the exhausted state of T cells.
Sorry, I couldn't hear that. Can you say it again?
If you have any experience where blocking PD1 or SLAM, if it is possible to revert the exhausted state of T cells.
We haven't done that ourselves, but that is what people do when they're trying to help people who have cancer. So that's how it works in cancer. That's what happens. It turns down these exhaustion markers to rejuvenate those exhausted T cells, and these drugs, they're called checkpoint inhibitors, are ones that are effective in some cancers to help patients battle against their tumor cells. But these have not been tested in ME as far as I know.